Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start a topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with alpha oxidation. In today's session, we will continue with some important organic acidureas. Then we have omega oxidation, in which dicarboxylic acidureas are seen. Then lastly, we have de novo synthesis of fatty acids, where fatty acid synthesis is a multi-enzyme complex. Good morning everyone. Let us start the topic. Here is a table where some important organic acidureas are seen. Example, methylmalonic aciduria, propionic acidemia, medium chain acyl-CoA dehydrogenase, long chain acyl-CoA dehydrogenase deficiency, and glutaric acid urea. Coming to Refsum species, it is a metabolic error due to the lack of alpha hydroxylase, that is phytanic acid oxidase, so that alpha oxidation does not occur and phytanic acid accumulates in the tissues. The patients present with severe neurological symptoms, polyneuropathy, retinitis pigmentosa, nerve deafness, and cerebellar ataxia. Regression of symptoms are observed with restricted dietary intake of phytanic acid. Milk is a good source of phytanic acid, which may be avoided. Coming to infantile refsum species, it is a paroxysmal disorder similar to Zellweger syndrome or adrenoleukodystrophy. Hence, phytanic acid accumulates along with very long chain fatty acids. Children do not survive long. Coming to omega oxidation, it is a minor pathway taking place in microsomes with the help of hydroxylase enzymes involving NADPH and cytochrome P450. The methyl group is converted into methyl alcohol and subsequently oxidized with the help of NAD plus to a COOH group to produce dicarboxylic acid. Omega oxidation becomes important when beta oxidation is defective and dicarboxylic acid, that is 6 carbon and 8 carbon containing acids, are excreted in urine causing dicarboxylic aciduria. Coming to de novo synthesis of fatty acid. In this table, a classification is given between beta oxidation and fatty acid synthesis. Site. In case of beta oxidation, the site is mitochondria, whereas in fatty acid synthesis, it is cytoplasm. Intermediates. In case of beta oxidation, it is present as CoA or coenzyme A derivatives, whereas in fatty acid synthesis, it is covalently linked to SH group of acyl carrier protein. In enzymes, in case of beta oxidation, it is present as independent protein, whereas in fatty acid synthesis, it is multi-enzyme complex. Sequential units. In case of beta oxidation, the 2 carbon units are split off as acetyl-CoA, whereas in fatty acid synthesis, 2 carbon units are added as 3 carbon malonyl-CoA. Coenzymes. In case of beta oxidation, it is NAD plus and FAD are reduced, whereas in fatty acid synthesis, NADPH is used as reducing power. Fatty acids synthesized mainly by a de novo synthetic pathway operating in the cytoplasm. So it is referred to as extramitochondrial or cytoplasmic fatty acid synthesis system. The major fatty acid synthesized de novo is palmitic acid, that is the 16 carbon containing saturated fatty acids. The process occurs in liver, adipose tissue, kidney, brain, and mammary glands. Coming to transport of acetyl-CoA to cytoplasm. The starting material for de novo synthesis is acetyl-CoA. It is formed inside the mitochondria from pyruvate. The inner membrane is not freely permeable to acetyl-CoA. Hence, the acetyl-CoA These units are delivered to the cytoplasm as citrate. Here is a diagram showing transfer of acetyl-CoA. In the mitochondria, acetyl-CoA and oxaloacetate combine together to form citrate in the presence of enzyme citrate synthetase. Then this citrate is transported outside into the cytoplasm, where it is broken down into acetyl-CoA and oxaloacetate in the presence of enzyme ATP citrate lyase. Then in the cytoplasm, oxaloacetate is converted back into malate in the presence of enzyme malate dehydrogenase. This malate is transferred into the mitochondria, and the cycle is repeated. Citrate is transported from mitochondria by a tricarboxylic acid transporter. In the cytoplasm, citrate is broken down into oxaloacetate plus acetyl-CoA. The enzyme is ATP citrate lyase. The oxaloacetate can return to mitochondria as malate or pyruvate. The system the system exists as a multi-enzyme complex. The enzymes form a dimer with the identical subunits. Each subunit of the complex is organized into 3 domains and 7 enzymes. In this diagram, we can see fatty acid synthase complex, which is divided into 3 domains. The first domain contains ketoacyl synthase, acetyl transacylase, alanine transacylase. Then the second domain contains hydratase, enol reductase, ketoacyl reductase, and acyl carrier protein. And the third domain includes thioesterase. Coming to advantages of multi-enzyme complex. Intermediates of the reaction can easily interact with the active sites of the enzymes. One gene codes all the enzymes, so all the enzymes are in equimolecular concentration. So the efficiency of the process is enhanced. Coming to first domain or condensing unit, it is the initial binding site. The enzymes involved are beta-ketoacyl synthase or condensing enzyme, acetyl transferase, and maronyl transacylase. Second domain or reduction unit, it contains dehydratase, enol reductase, beta-ketoacyl reductase, and acyl carrier protein. The acyl carrier protein is a polypeptide chain having a phosphopantothene group to which the acyl group are attached to in the thioester linkage. Coming to third domain or releasing unit, so ACP acts like a coenzyme carrying fatty acid group. It is involved in the release of the palmitate synthesized. It contains thioesterase or deacylase. Coming to step 1, carboxylation of fatty acyl-CoA. The first step in the fatty acid synthesis is the carboxylation of acetyl-CoA to maronyl-CoA. The acetyl-CoA carboxylase is not a part of the multi-enzyme complex, but it is the rate-limiting enzyme. Iodine is a member of B-complex vitamins. It is necessary for this reaction. In the diagram, we can see carboxylation of acetyl-CoA. Acetyl-CoA plus carbon dioxide gives maronyl-CoA. In the presence of enzyme acetyl-CoA carboxylase and conversion of ATP to ADP, and the presence of biotin. The enzyme is allosterically regulated, the major effectors being citrate, that is a positive regulator, and palmitoyl-CoA, that is a negative regulator. The reaction is similar to the carboxylation of pyruvate into oxaloacetate. The elongation of the fatty acids occurs by addition of 2 carbon atoms at a time. The acetyl transacylase catalyzes the transfer of the acetyl group, that is two carbons, to a cysteine SH group of the condensing enzyme of the other monomer of the fatty acid synthase complex. Acyl carrier, it is written as acetyl coenzyme A plus condensing enzyme gives acetyl condensing enzyme plus coenzyme A in the presence of acetyl transacylase. Next step is carbon 3 carbon and 2 carbon units are added. One molecule of acetyl CoA containing 2 carbon and one molecule of malonyl CoA containing 3 carbon bind to the multi-enzyme complex. Malonyl transacylase transfers the malonyl group to the SH group of ACP of the monomer of the enzyme. It is written as malonyl coenzyme A plus acyl carrier protein gives malonyl acyl carrier protein plus coenzyme A in the presence of enzyme malonyl transacylase. The acetyl and malonyl units are condensed to form beta-ketoacyl ACP or the acetoacetyl ACP. During this process, one carbon is lost as CO2. The enzyme is called condensing enzyme or ketoacyl synthetase. Here we can see that acetyl condensing enzyme plus malonyl acyl carrier protein together they form acetoacetyl ACP. In the presence of enzyme, condensing enzyme or ketoacyl synthase, and the removal of CO2 and condensing enzyme. Step 4 is reduction. The acetoacetyl ACP is reduced by NADPH dependent beta ketoacyl reductase to form beta hydroxy fatty acyl ACP. In the diagram, it is written as acetoacetyl ACP converts into beta hydroxybutyric ACP in the presence of enzyme ketoacyl reductase and conversion of NADPH to NADP+. Step 5 is dehydration. It is then dehydrated by dehydratase to acyl ACP, otherwise known as alpha beta unsaturated acyl ACP. Step 6 is second reduction. The acyl ACP is then reduced by acyl reductase to utilizing a second molecule of NADPH to form butyryl ACP. Here in this diagram we can see that beta hydroxybutyryl ACP is converted into acyl ACP in the presence of enzyme dehydratase and removal of H1H2O. Then this acyl ACP is converted into butyryl ACP with the presence of enzyme acyl reductase and the conversion of NADPH to NADP+. Here ends our session. Kindly like, share and subscribe the channel. Thank you.